Okay, so we will start our transfection experiment today. So today's experiment, we will include two steps. Okay, step one, we wanted to prepare DNA in a transfection agent mixture based on this table. Okay, and since we wanted to mix 100 microliter of the DNA and 2 microliter of the transfection reagent, okay, for the transfection reagent alone group, we are going to use 100 microliter of the medium to replace the DNA, okay? And for the negative group, okay, we don't add anything, but we will prepare 100 microliter of the medium to use as the negative group, okay? Okay, so now we prepare the DNA transfection reagent mixture. So don't forget to spray alcohol on your gloves. Okay, we already transferred the micropipettes and the medium only, and also some tube rocks in a different pipette tips box into the cell culture food. And also we need to use Eppendorf tubes. Okay, here are our autoclaved Eppendorf tubes. Okay, so we needed to have 4 groups. Okay, in the lab notebook I put 5 groups. Okay, but today we only use the 4 groups. There is one duplicated group. Okay, we will not duplicate it today. Okay, and first label the tube DL. D, which means DNA only. L means the DNA plus LTX reagent. L only and a negative. Since our DNA is a stock DNA, so we calculated how much volume in order to get 2.5 nanogram per microliter for 100 microliter, and we calculated we only need 1 microliter. So what we are going to do is to mix 1 microliter DNA stock and a 2 microliter LTX reagent and a 97 microliter of the medium for the first two. And when you take a small volume from a big bottle, okay, so I don't want you to insert the whole, pipette into the bottle. Okay, I want you to tilt, the bottle, and only put the sterile micropipette tips into the bottle, and then take 97 out, microliter out. Okay, to save your time. So for the second tube, we need 99 microliter of the medium. So you don't need to change the tip, just adjust the scale to 99. Okay, and then take 99 microliter, put it into the second tube. And for the third one, we need 100 microliter of the medium and 2 microliter of the LTX reagent. So you adjust to 100 and take 100 out. And for the last one, we need 100 2 2 microliter of the medium. Okay, adjust 102. Okay, so now we are going to add a 2 microliter LTX reagent to the first tube. So this is the 2 microliter pipettes. Okay, so this is our LTX reagent. Okay, remember to open the cap inside the hole. Okay. Okay, make sure you add the small volume under the surface of the liquid. And then pipe it up and down for several times. Okay, also we need to add 1 microliter of the DNA. Okay, I forgot to add 1 microliter DNA, and we first added the LTX reagent. That's fine. So now I'm going to add 1 microliter of the DNA. Okay. Okay, so this is our DNA stock with the relatively high concentration. Mm-hmm. Okay. One microliter. Okay, this will be the first tube, which contains DNA and a cationic lipid reagent. Okay. For the second one, it only contains DNA, so we are going to add 1 microliter of the DNA to the 99 microliter of the medium. Okay, 1 microliter DNA. So this will be the DNA only group. For the third one, we need to add 2 microliter LTX reagent. Okay, adjust it to 2 microliter. Okay, this is our LTX reagent. Okay, this tube doesn't have DNA. Okay, only has LTX reagent. Okay, last one, we are not going to add DNA or LTX reagent. Okay, we are going to keep the tubes at room temperature for 30 minutes to allow it, to form the complexes. So these are the cells, we subcultured 2 days ago. It is now ready to do the transfection. Okay, so what we wanted to do is to change the medium. Okay, we want to remove the old medium and put the fresh new medium, and then add the transfection, DNA mixture. Okay. Okay, so we are going to remove the old medium.
After discard of the old medium, we can add 1 milliliter of the fresh medium to the dish. Make sure you add the medium slowly against the side of the dish. Otherwise, you will disrupt the single layer at the bottom of the dish. After replacing all of the medium with fresh medium, we can label 4 dishes with the name of those 4 different groups. I will start from DL, and next is D, and L, and negative. I will now add the corresponding mixture to the corresponding dish. Make sure you adjust the pipette to 100 microliters. And make sure you add the mixture dropwise. And gently spread the mixture in the medium. Do the same to the rest 3 tubes.
We only do one complete set, okay, for the entire class to use. Okay, we are not going to do 20 or 30 for each of you to use. Okay, we only make one complete set. And Wednesday, we are going to take the fluorescence image, and we are going to upload the image so every group can download and use. Okay. And for the Western blotting preparation, we are going to just use the first group and also the last group to prepare positive and negative transfection groups. Okay, so we will prepare for the whole class to use. Okay, so we already calculated how much we need to mix. Okay, so we are going to mix. It's around 2.5 milliliter of the medium and 17.4 microliter of the DNA and 50 microliter LTX reagent, okay, to prepare transfection positive groups, okay, and then we just prepare the same total volume of the medium for the negative transfection groups, okay. So now we need 2 groups, 2 tubes. Okay, and one is positive, the other is negative group. Positive and the negative group. Okay, we need 2.5 milliliter of the medium. Okay, this is 1 milliliter, 2 milliliter, and we need 0.5 milliliter. Okay, for the negative group, we only need 2.5. We also need 2.5 milliliter. So we first add 0.5 and then 2 milliliter. Okay, and in the first tube, we need to add 17.4 microliter of the DNA. Adjust to 17.4. We need to add DNA stock. You can also tilt. Okay, try not to insert the whole pipette into the tube. Okay, tilt to get to the solution and then add it inside. Okay. And we need to add 50 microliter LTX reagent to the first tube. Okay, we will do the same thing, tilt a little bit, so we are able to add the LTX reagent into the solution. So now do a gentle mix and keep at room temperature for 30 minutes.
Okay, so after replace the old medium, and I label 22 dishes with a positive sign. That means they are transfected with the plasmid and the transfection reagent. So what I'm gonna do is add the positive reagent with DNA and also the transfected reagent, 100 microliters to each dish. So for those dishes that got the negative group, we label them as a negative sign on the top of the dish, and then do the same way, add 100 microliters of the mixture from the negative tube. After adding all the reagents to all the dishes, we then transfer them back to the incubator for incubation for 24 hours.